expert, Professor Sanjay V. Malhotra from Oregon Health and Science University. He will be presenting on molecular imaging in drug development with a focus on introduction to multimodality molecular imaging. This session will take us into the exciting world where chemistry meets imaging and innovation.
Thank you, Tanya. And my apology, I will have to change a little bit again. In order to do this, I will go over on pharmacology and toxicology, and then I will bring to imaging. So, but imaging is coming. Stay tuned. All right, good morning. आप लोगों को आराम तो मिल रहा है ना कि मेरे लेक्चर सुनने के बाद में इतना बोरिंग हो जाता है या इतना ऊपर से भारी हो जाता है? ठीक है. No, so what as I was saying कि so you saw we learned about the कहाँ से शुरू हुआ, किस तरह से शुरू हुआ, जो chemical looking from the lens of chemistry towards the biology. एक बायोलॉजी को देखना है तो केमिस्ट्री के लेंस से, कि केमिकल्स के लेंस से उसे देखने की कोशिश करें. So we saw chemical solutions for biological. We looked at the biological space, but the focus is experimental therapeutics, drugs. So I showed you कि, can you hear me? Yeah. When you look at, this is just showing you a few-year-old numbers. Okay, if you have Yeah, no, this works. मैं चलते हुए बात कर रहा था तो okay, it's working now. So as I was telling, and as you saw today, as to develop a drug, to discover a drug, it's a multidisciplinary enterprise. Chemistry, biology के अंदर भी आप देखें pharmacology, toxicology, disease biology, understanding what is immunology, understanding how assays are developed, understanding how models are developed, if there are models are right or wrong, in vitro models, cell line models, animal models, and even within the animal, what type of animal models you want to create, and then PDX, patient-derived xenograft models, ex vivo human models, taking the human samples and creating a model. Each one of these is an expertise. Is a discipline. उस सब के बावजूद you have to juggle all of these things to make sure that the drug that you design, the things that you design, has the effect that you are looking for without causing any toxicity or damaging. ऐसा नहीं है कि एक चीज़ से तो ठीक कर दे और दूसरे से मार दे. So इन सारी चीज़ों को, and I'll show you how multiple you are juggling multiple of these things together. So and therefore, in order to bring one good drug to the market, this chart, as you start with, for potential new medicine, just the basic research. You find out discovery. You do preclinical, all the things that I'm telling you, all the preclinical part, and I'll just show you briefly about the clinical side. When you do, then you start phase 1, phase 2, phase 3 clinical trials, and you do not your drug may fail because of it has a property which is not suitable for human consumption, and it may just drop out. So very lengthy, highly resource intensive, multidisciplinary and long process. So if you think, for example, if there are several thousand candidates, molecules which are being tested, maybe only 4 or 5 will enter the clinical trial, and maybe 1 will enter into the commercial world, which will be approved by that. So it's a very, and why is that it takes long time? So just look at, here, for example, so drugs could fail for multiple reasons, and I have tried to give here, for example, so this is this is maybe a little bit still an old data, but look at this. 39% of the drugs fail because their PK, their pharmacokinetics properties are not good. So when they get to the point of the clinical trial, they fail, or something happens, they fail. 30% of the time, there could be lack of efficacy. What you see, the drug is effective. Drug is effective in your Petri dish model, in vitro model. You can take into animal model, it may be effective, but when it comes to human, you don't see the same effect, whatever be the reason. So 30% of the drug fail because of that, because what you see, the properties which are working in ex vivo human models, they don't pan out in the clinical trials, or so drugs fail. And you see there are various other, the animal toxicity, they are very toxic, they don't go further. And then 5% could be miscellaneous reason, adverse effect, you have many side effects which you are not expecting, or there could be 5% with the commercial reasons, but there could be many reasons. So even though you start with the hope that research part, that drugs are working, but when it gets to the humans, the results are not the same. Now, among many of these reasons, one of the reasons where these things do not pan out, and as you see here on the left is the distribution, for example. Are they so let me take to the next slide. Look here, for example, you take, so I briefly mentioned yesterday, but let's, for example, you take an oral drug. You take a drug, it has to be absorbed in the intestine, right? Because it has to be absorbed, then it has to be distributed throughout the body. And to reach, and somebody asked me yesterday, how does it reach to the place where it's supposed to be, right? So it has to, the process, the mechanism, the route of delivery should be such that it reaches to the intended place that you are interested in. So and see, this is basically what happens. So you take a drug, it goes to stomach, as I mentioned, oral, in the case of oral, it goes to the stomach, because of its acidity, it breaks down, and then it gets into the plasma and it gets distributed. And in there, as I showed yesterday, the pH is variation, a lot of variation of pH, and if you have a small molecule, its property has to match with that. It has to be soluble, it has to be bioavailable, biologically available to the site of interest. If it is not available, you will not see the drug effect, because वहाँ तक वो पहुँचा ही नहीं है जो अपने action कर सके. So what happened? It enters so liver enzymes, they metabolize, they break it down to, because those enzymes break it down, the food that we eat, in this case, for example, the drug, and then elimination. Some metabolites which will go this way into the distribution, into the plasma protein, into the bloodstream, and then it gets distributed throughout. This is the distribution process. So this is a very simplified way. Now, this is the oral route. IV, many drugs are not suitable to be taken orally because their properties are such they cannot be, I mean, if you take, they will break it down, so जो उसका active ingredient है वो intact नहीं रहता है. Then either they are given to the bloodstream or given to IV. So through the IV, this route, for example, IV में जब देते हैं, it enters into the plasma, blood, and it gets distributed very quickly. And IV के अंदर जब देना होता है, so see key points I'm telling you, and I'll connect in my subsequent slides. Goal is कि drug has to reach to the site of interest. अगर वहाँ पहुँचा नहीं तो उसका असर नहीं होगा. Now if you give the oral route, it gets metabolite, it breaks down, goes to the various part of the body. Various parts, they have different functions because there are hundreds and thousands of enzymes जो catalytic reaction कर रहे हैं उस molecule के ऊपर, and it gets distributed or it gets cleared, elimination process gets cleared through whichever route. IV, so oral route के अंदर usually the concentration of the active drug is higher. IV is highly effective. IV के अंदर डोज कम हो तो भी क्योंकि वो काफ़ी है to reach the sites, because it gets into the plasma, gets into the blood, and blood circulates throughout the body. It gets circulated पूरी body के अंदर. So therefore the reason I'm mentioning is, if you next time if you see कि oral pill का size है इतना है, उसमें पढ़ो कि active ingredient कितने हैं, right? और दूसरा, when you see the same match on IV के अंदर जो दिया जाता है कितना है, तो usually the concentration will be smaller, because small concentrations is enough to reach the site of action. Oral में लेके उसका काफ़ी हिस्सा तो eliminate हो जाता है because of the enzymatic reaction that happened there. So understanding how all of these things, so how the drug has to be absorbed, it has to be distributed, it has to be metabolite, break down to metabolites, pieces that reaches to the site of where action where it needs to be, and then जब हो गया उसका function, जो उसके metabolite आगे बने, they are eliminated. So the process ADME, absorption, distribution, metabolism, and excretion. ADME is an extremely important subject, and for chemists it's relatively easier to understand compared to biology. Because what you are doing is basically the physiochemical properties of the molecule and what mechanistically they will be break down in various parts of the body. वो अगर absorb हो रहे हैं तो उसकी physiochemical property कि they get absorbed. If they are distributed, then they are distributed through some mechanism कि they survive through that mechanism. ऐसा नहीं कि टूट गए, they break down, the molecule is broken down. And then metabolites, वो metabolites अगर उनके टूटते हैं तो क्या बनता है उससे. So all of these things, and elimination or clearance, they get eliminated. So all of these are relatively easier for a chemist to understand, because it's all around physiochemical properties. You design your drug in such a way, you look at the physiochemical properties of the drug, you can manipulate, you can play around with this thing. So this is what, quote unquote, is the field of pharmacology. So pharmacology basically is the science of how drugs and biologics act on biological system and how the system responds to it. Okay. And clinical pharmacology basically is when it's the clinic, is basically applies to the basic science and principles of pharmacology together. So when you look at pharmacology, it is your basic scientist is looking at from the concept of trying to understand what the drug will do, how it will do, and try to manipulate the drug in order to discover good thing, and then translating that information to clinical side. So think for a second, आपने ये सुना होगा सेम कि कई चीज़ें जो हैं ओरल दी जा रही हैं, कई जो हैं डर्म से दी जा रही हैं, राइट, स्किन लोशन जो दी जा रहा है, कई चीज़ें जो हैं आईवी से दी जा रही हैं, इंजेक्शन से दी जा रही हैं मसल्स के अंदर, राइट. Ask yourself why. सबको ओरल क्यों नहीं दे देते, या सबको आईवी क्यों नहीं दे देते, राइट? Simple. Why? It's based on the properties. Based on the properties of what you're, and multiple things. One is the property, and one how quickly and it reaches. Now I will come to that, and I was going to mention a point, but I'll come back to that. So basically, experimental pharmacology, it's preclinical study. Can you take care of understand how again ADME talks, absorption, distribution, metabolism, and excretion, how it happens in various of these models, and clinical pharmacology inside the clinic किस तरह से किया जाता है कि how the drug will behave, how whatever the candidates will behave. So clinical study is done. So that's why when you hear phase 1, phase 2, phase 3 की जाती है study, I will come to that, what the difference between those phases of studies are. But clinical studies are done in order to first make sure that it is performing as intended before FDA or any regulatory agencies approves and वो masses को easily commercially available हो. So that part of the study is basically what is. So again, I'm giving you this thing again and again just to hammer the definition and understanding of it. So pharmacology again is the science of drug and medication. Specifically, it is the interaction of, so I will come to that, how body reacts, what body does to the drug or medicine, and the second thing is what drug does to the body. So pharmacokinetics and pharmacodynamics, two different terms. आपने ड्रग लिया, ड्रग ने जाके बॉडी के अंदर क्या किया, और आपकी बॉडी के अंदर जो एंजाइम्स या बॉडी के अंदर उसने ड्रग को क्या किया, दोनों के अंदर. सो that's the difference between the two. So if you look at, there are multiple steps. Again, specific, again, the rate that a chemical is absorbed or distributed, understanding of that, that's again pharmacokinetic. The rate and pathway of drug metabolism. अगर वो जाते हैं लिवर में, किस स्पीड से जाते हैं और क्या मेटाबोलाइट्स बनते हैं, किस रेट से बनते हैं, किस रीत से वो डिस्ट्रीब्यूट होते हैं, अंडरस्टैंडिंग ऑफ दैट. एंड अगेन द प्लाज्मा कॉन्सेंट्रेशन ऑफ ड्रग over the time. And this is again, when I say over the time, can anybody think of why I'm saying over the time? Drug plasma distribution kitne time mein hota hai, over the time how it gets distributed, why is that important? So first कीजिए, ड्रग लिया, ले तो लिया, मगर उसकी कॉन्सेंट्रेशन जो है, whether through oral route or through plasma or the IV route. Let me step back. कि ड्रग जो है 5 मिलीग्राम पर किलोग्राम बॉडी वेट चाहिए उसका असर करने के लिए IC50, if the concentration which will kill the 50% of, has the 50%, that's IC50. So if you take the drug, if a person takes the drug and required concentration is, next time आप जब देखें, just flip the, नहीं मैं देखा, मैं दवाई नहीं निकाल रहा, मैं सोचा था कि मेरे पास में दवाई है तो आपको दिखाऊं, कि you will see, I mean, this is again, it will automatically, you are self-educating, because even when I take a food, I like to read and see what the ingredients are, always. ये आदत पड़ गई है. I look at the ingredients, चाहे food के अंदर भी ingredients क्या हैं उसके, कितने % carbohydrate है, कितने % protein है, just as a chemist, I always look at that. ये है कि पराठों पे लिखा नहीं होता, नहीं तो मैं पराठा भी पढ़ूँ मैं क्या हुआ है. But point being, that connects you कि इसके ingredient क्या हैं, कितने carbohydrate, protein, that's one part. Anything that you take, even I just go and buy shaving cream, I look at back कि उसके अंदर कौन से menthol कितना है और वो सब क्या है. It just as a chemist, मेरे को आदत पड़ चुकी है. Drugs अगर आप देखें, कोई भी आपको दवाई का पत्ता मिलता है, just turn it around and see उसमें कौन से ingredient कितने % है, right? So let's say if the drug for its intended effect is 5 mg per kg, 5 milligram per kilogram body weight चाहिए उसका, बट उसकी प्रॉपर्टीज ऐसी है कि ड्रग लिया, वो सॉल्युबलाइज नहीं हुआ, और मेजॉरिटी ऑफ ड्रग है वो आपका किसी भी रूट से वो बाहर निकल गया, उसका असर होगा नहीं होगा. 1 पॉइंट. सेकंड, तो उसमें अगर 50-90% निकल गया तो 10% बचा. अब 5 mg पर किग अगर उसका चाहिए और आपने 5 mg पर किग लिया तो उसमें से 90% तो निकल गया बाहर, वो क्लियरेंस हो गया, यूरिन रूट से, जिस रूट से वो बाहर निकल गया. 10% बचा तो उसका इंटेंडेड से होगा नहीं. फिर लेंगे तो बोलेंगे जी दवाई काम नहीं कर रही. इट्स नॉट द दवाई काम नहीं कर रही, बट एक्चुअली मार्केट तक आता नहीं है. बट दैट्स कि मार्केट, दैट्स हाउ दीज थिंग्स आर स्टडीड. Second thing, for a second, okay, that is one part. Second thing, okay, let me ask you this question. आपने सुना होगा कि डॉक्टर से आपने ली दवाई, वो कह जी कि आप दिन में 2 बार लीजिए या दिन में 3 बार लीजिए या एवरी अदर डे लीजिए. Why? Think, क्यों? Why एक पिल को 3 बार क्यों लेना पड़ रहा है? या एक ही बार क्यों लेना पड़ता है, ऑल्टरनेट डे पे क्यों लेना पड़ रहा है? राइट, सिंपल क्वेश्चन. आई एम श्योर आप लोगों ने सुना हुआ ये, take twice a day, 3 times a day, or take these many pills or anything. As a chemist, convert that back. Why? So now you see, it will be good to read the ingredient. It will be good to read कि उसके active ingredient क्या है. And so here is the reason. Drug की half life जो होती है ना, clearance half life, कितना जो पहुंचता है. So if उसकी half life के basis पे doses उसके decide किए जाते हैं. If you take the drug, how long it will stay inside the plasma, inside your body? 50% of the part. उसका 50% कम होता जाएगा, उसका 50% कम होता जाएगा, right? So मगर if drug जो आप Tylenol, well I don't know Tylenol में यहाँ पे है acetaminophen, I don't know कौन से ब्रांड नाम से यहाँ बिकती है, not एनासिन के अंदर तो डिफरेंट होता है, बट सिर की दवा, सिर दर्द की दवाई फॉर एग्जांपल, राइट? सो उसका जो है हाफ लाइफ जो है प्लाज्मा रहने के बाद में 50% रहेगा. अगर 1 ग्राम लिया, 1 ग्राम इज़ टू हाई, भला से 100 मिलीग्राम लिया, उसका अगर 6 घंटे के अंदर 50% उसका क्लियरेंस हो गया, तो बाकी आज 6 घंटे बाद में उतना ही बचेगा, कम बचेगा. उसके 6 घंटे बाद में और भी उसका भी 50% कम हो जाएगा, राइट? तो मगर बॉडी को उसका इफेक्ट करने के लिए आपको पूरा 100 मिलीग्राम ही चाहिए. एंड दैट्स व्हाई डॉक्टर से एवरी 6 आवर्स कि आपकी बॉडी के अंदर जो है उसकी कॉन्सेंट्रेशन मेंटेन रहे, क्योंकि उसका क्लियरेंस जो होता जाता है, बॉडी गेट्स क्लियर. सो दैट इज़ हाउ जो डोजेस के बेसिस पे आपको डॉक्टर 2 बार लो या 3 बार लो. अगर डॉक्टर के इसको इतनी बार लो, उसमें घबराने की जरूरत नहीं है. नाउ एज ए केमिस्ट नेक्स्ट टाइम व्हेन यू हियर, उसको आपको बताना चाहिए कि जस्ट टू मेंटेन द कॉन्सेंट्रेशन इनसाइड द बॉडी. डॉक्टर साहब एक ही बार में क्यों नहीं खत्म करते, 3 बार मैंने क्यों लेनी है दवाई? डॉक्टर क्योंकि उसका प्रॉपर्टी ऐसी है कि वो क्लियर होता जाएगा. ड्रग अगर क्लियर होता गया बॉडी में नहीं, बॉडी में नहीं रहा तो उसका फर्क असर महसूस नहीं होगा. सो दैट्स हाउ दीज थिंग्स आर डिजाइन. सो एडमी टॉक्स, नाउ यू कैन सी एज ए केमिस्ट इज relatively easier to get into that field, understand and be able to master that field, क्योंकि it's all around physiochemical properties. And as a chemist, if you look at the structures of the molecule, उसी के जैसे मैंने स्ट्रक्चर दिखाया था the periodic table, these are all atoms. उनके कितने इलेक्ट्रॉन्स है, कितनी आयनिक पोटेंशियल किसकी क्या है, वो you can draw all of these. So physiochemical कितने उसके अंदर कौन से functional groups हैं, they are reactive functional groups or not, all of these because they are interacting with enzymes. So anyway, long story short, that's the basis of how doses क्या हैं, कितने टाइम लेना है, whether oral route, whether IV route, those are the things that are decided based on ADME talks. So if you look at just the pharmacology areas हैं, ड्रग कॉम्पोजिशन क्या है, उसकी प्रॉपर्टीज क्या है, ड्रग का सोर्स क्या है, इज इट अ सॉलिड और इज ए लिक्विड जो आपको दिया जा रहा है, आईवी के सोर्स पे या कहीं भी दवाई भी ली जाए. सो ड्रग अगेन, ड्रग डिजाइन, दिस इज अगेन कि उसकी प्रॉपर्टीज क्या है, इंटरैक्शंस, व्हेन द ड्रग गोज इनसाइड, how it is interacting with enzymes, or if there are multiple drug, people are taking multiple drugs, how there is drug-drug interaction. Pharmacokinetic clearance of 50% availability of one drug versus if multiple drugs are, देखें दूसरे का असर कितना होगा. कई बार होता है ना, वो कहते हैं कि डॉक्टर अच्छा ये वाली दवाई भी लो और साथ में ये वाली दवाई भी लो, और मे बी समटाइम मल्टीपल दवाई दे, तो वो उसको बैलेंस करने की कोशिश करे. सो दैट्स द क्लिनिकल फार्माकोलॉजी. उसको बैलेंस करने की कोशिश करे कि ये दवाई इतना असर करेगी, उसके साथ में आप दूसरी दवाई लेंगे तो उसके ऊपर क्या इफेक्ट करेगी. बट ऑल दीज डॉक्टर के यहाँ पहुँचने से पहले प्री क्लिनिकल बेसिक साइंटिस्ट like yourself, they do all the discovery part. Doctor only implements. That's why clinical trials happen, to do that. But see, within pharmacology, kitni interaction, chemical biology, as I've been talking, molecular and cellular metabolite banenge, how distribution will happen, excretion will happen. These are all biological mechanisms. Organ systems is working on antipathic capabilities, signal transduction, medical application, therapy, all of these are come under pharmacology. So, as I mentioned earlier, pharmacology is combination of pharmacokinetics and pharmacodynamics. pharmacokinetics, as you see, to study how an organism affects the drug, what body does to the drug. क्योंकि बॉडी के अंदर एंजाइम्स हैं, ड्रग जब अंदर आया तो उसने उस बॉडी ने क्या किया, उसके ऊपर तोड़ मोड़ के कैसे मेटाबोलाइट उसका बनाया, उसका उल्टा कि ड्रग ने बॉडी के ऊपर क्या असर किया. दैट्स फार्माकोडायनामिक्स, द स्टडी ऑफ हाउ ड्रग अफेक्ट्स द ऑर्गेनिज्म. एंड नाउ यू कैन अप्रिशिएट कि दोनों में फर्क है, क्योंकि बॉडी के अंदर आपके जो एंजाइम्स है वो उसके ऊपर अटैक करें, नॉट अटैक बट दे विल इंटरैक्ट, दे विल डू केमिस्ट्री देयर, राइट. Drug comes in with its own property inside the body, so drug is trying to do something in there. So उस पार्ट को समझना कि बॉडी के ने ड्रग के ऊपर क्या किया, दैट्स वाई हाफ लाइफ अगर उसकी है ड्रग की, दैट्स बिकॉज बॉडी उसको तोड़ रही है, उसको क्लियर कर रही है. सो दैट्स फार्माकोकाइनेटिक्स. व्हाट ड्रग डज टू द बॉडी इज फार्माकोडायनामिक्स. So the understanding of each one of those is extremely important when you design a drug to see कि किस तरह से उसके डोजिंग रेजिमेंट बनेंगे, क्या प्रोटोकॉल्स बनेंगे. See, okay, I have this method. I didn't realize I already have that. Both together influence the dosing. What benefit will be there? What will be the adverse effect? वो कहते हैं ना कि ये कुछ यहाँ पर साथ में स्ट्रॉन्ग दवाई या साथ में नौजिया भी हो जाता है. सुना होगा आपने ये सारे टर्म जो है, these are common terms. थोड़ा होता है मगर उसके कारण थोड़ी नौजिया होता है, उल्टी भी आ जाती है, बट ड्रग इज इफेक्टिव बिकॉज दैट्स द साइड इफेक्ट, राइट. बट देन यू बैलेंस के यू नो कि बेनिफिट वर्सेस द साइड इफेक्ट. द साइड इफेक्ट शुड नॉट बी ओवर टेक ओवर द बेनिफिट साइड ऑफ इट. सो दैट बैलेंसिंग इज व्हाट द साइंटिस्ट डज. दैट बैलेंसिंग इज व्हाट ए फार्माकोलॉजिस्ट डज. ट्राई टू फिगर आउट what is the fruitful balance which allows for drug to come out, to be move forward for preclinical से clinical trial से लेके commercial उसके अंदर, so that intended use is humans. अगर humans के लिए drug बन रहा है, intended use is humans, so they can benefit from that. And that's why जो पहले मैंने दिखाई है, that's why it takes investment. Just imagine, पहले तो screen करना, as I was telling you yesterday, you do high throughput screen, you take thousands and thousands of compounds, now millions of compounds, and with in silico you can start with even billions of compounds to design computationally and bring it down to an actually 10,000 or several thousand. I mean, we have in our collection million, 2.25 lakh करीब हमारे पास में है compounds हैं that we never needed, we screen those. पर करते करते जब यहाँ तक पहुँचा, उसके बाद में वहाँ से लेके जाने के अंदर इट्स एन एक्सपेंसिव प्रोसेस एंड हाईली साइंटिफिकली रिगर चाहिए होता है, क्योंकि आफ्टर ऑल इट्स फॉर ह्यूमन यूज. इट हैज़ टू बी डन प्रॉपर्ली. यू कैन नॉट मेक ह्यूमन एज ए गिनी पिग कि उनके ऊपर ट्रायल करते हैं, चलो ये काम नहीं किया दूसरे करें, पर जिनके ऊपर किया उनका बेचारों का क्या होगा, राइट. सो दैट्स वाई इट्स ए लेंथी प्रोसेस, इट्स एन एक्सपेंसिव प्रोसेस, एंड इट्स ए हाईली interdisciplinary process where many disciplines come together. So this is basically what is a field in itself. ADME talks comes with it, toxicology, and I'll cover a little bit of that too. But this is what is the absorption, the process of a drug entering into the system, circulation from the site of administration. So site of administration means it could be oral, it could be IV drug, it could be dermal. उस साइट से लेने के बाद में वो किस तरह से एब्जॉर्ब होता है, दैट्स एब्जॉर्प्शन पार्ट. सेम वे लुक एट द डिस्ट्रीब्यूशन, द हाउ इट इज disseminated, how it is distributed throughout the body. And then metabolites, how in the liver, for example, how liver enzymes, they break it down, क्या metabolites बनते हैं, understanding of that, it's a huge field by itself. Each one of these is a field, is a scientific field. And then excretion, how whatever is left over, how it gets cleared. क्योंकि अगर वो क्लियर नहीं हुआ, बॉडी के अंदर ये accumulate होता हुआ करते तो वो टॉक्स साइड इफेक्ट्स करेगा, टॉक्स करेगा. तो how it gets broken down by the enzymes and how it gets cleared. Each one of these understanding is a lengthy process. So I can go over, I'll go over one or two, and then I have slides which you'll get to. So I will, at least you'll get the concept of, yeah. So let's talk about absorption. Now main routes, oral, dermal, IV, जो routes हैं, उसमें when through any one of these routes, if the drug is taken, how is it absorbed?